So we went back to the drawing board and we took a very conventional GelMA, so gelatin methacrylate, and we thought, okay, can we keep it printable or even maybe improve the shape fidelity, the quality of our prints, while also allowing cell migration at a very high level. So we achieved that by introducing some supramolecular units. So we put adamantane, these purple boxes here, together with cyclodextrin, which is what is drawn here as a bucket, which basically permit a guest host bond that is completely reversible. And the beauty of that is that basically it makes the gel macroscopically stiffer. So now if we print these wheels, if we make it with normal GelMA, 5% weight per volume, they collapse under their own weight. But with this material, they're able to withstand their own weight, and we can print them very nicely. But at the microscopic level, the cell level forces, cells are able to break these bonds with minimal integrin-mediated stresses, and therefore they can migrate. And after they move on, the gate closes back again, giving support to the structure. So basically, we can combine a beautifully suitable microscale environment for cell with the macroscale environment, which allow stiffness and printability. What you see here in green are endothelial cells that are populating a large print with very high migratory capacity.